I'm honored to introduce nephrologist and ASF Medical Advisory Committee member, Dr. James Simon. He'll serve as our moderator for the session on genetics and understanding your genetic test results this morning. Our other esteemed experts on the panel today are nephrologist and researcher, Dr. Andrzej Rastogi of UCLA, who also serves on our medical advisory committee, genetic counselor Mary Beth Roberts, who is co-founder and co-chair of the Renal Genetics Special Interest Group at the National Society of Genetic Counselors. You can read their full biographies in the agenda section in the app. You can read their full biographies. If you have not yet had the chance, please remember to watch the informative interview with Mary Beth by clicking on the genetics vid icon on your app at some point this weekend. But now it's time to turn our attention to our moderator and our panelists for questions and answers. So I encourage you to click the Q&A button on your screen to ask your valuable questions. And I'm going to turn everything over to Dr. Simon. Thank you all for being here. Dr. Simon.
Thank you, Lisa. Welcome, everybody. Appreciate you attending the ASF ACT meeting. This is a really great resource. I've had a lot of patients that have learned a lot from these meetings and really feeling part of the community. We're starting off with the basics of genetics today, and this is a Q&A session. And as Lisa said, if you do have some questions that you would like us to address in the next 54 minutes, please submit them in the Q&A section, not in the chat. And that's at the bottom of your screen, just left of center. So today we have Dr. Andrzej Rastogi. He's Chief of Nephrology at UCLA, and he's also on the Medical Advisory Committee for the Alport Syndrome Foundation, which is where I know him from. And then in addition, Mary Beth Roberts, who I work with quite a bit at Cleveland Clinic. She is one of our awesome renal genetic counselors. Really have a great resource here. She works with Dr. Xiaoling Wang, who's double board certified in nephrology and genetic medicine. And they're both going to serve as our resources, our experts today. I do have some pre-submitted questions that we either got from Facebook or at least provided from other sources. And then we'll start with those. And as other questions do come in, we'll add those in as well. So first, Andrzej, Mary Beth, I think we'll start with at the basics, and we need to walk a fine line of explaining the basics to everybody without getting too deep into it, but also trying to get to the point where some of our patients and family members do understand the basics and want to know a little bit more. So we'll start small, or we'll start basic and work our way up. And I think the first question is a good one, which is just what is a mutation? And do all Alport patients have a mutation? What do all the numbers mean in the report after the mutation? Mary Beth, you want to start with that?
Yeah, absolutely. So hi, everyone. I'm Mary Beth. I think when we ask the question, what is a mutation, it's really helpful to first quickly define what is a gene, because it will help to make everything else make a little bit more sense. So genes are the parts of our body that tell us how to develop and grow. So when there's a mutation, or what we call a pathogenic variant in a gene, it means that the message that gene carries is no longer working properly. And so whatever function that gene has in the body cannot then be carried out in the normal way. Not everybody with Alport syndrome has a genetic mutation that we can find. Most people do. But there are, there are some families out there who have all of the signs and symptoms. They have a biopsy that very clearly suggests Alport syndrome, and their genetic testing will come back normal. So in those cases, we still say that they have Alport syndrome, but it may be that we just can't find the mutation at that time.
Great. Yeah. So, Mary Beth, I think that was a great explanation. Just adding a few things to this pretty inclusive definition that you give. First of all, I do want to thank Alport Syndrome Foundation, Lisa, Kevin, and the entire organization for putting this together. This serves value to all my patients. Actually, some of them are actually on Tara, one of my patients who's on this as well. So we're very grateful. And I learned myself so much. This is a rare disease, and these kind of events are very, very important. We learn from each other. Now, for time's sake, I'll keep it very short and simple, but just going on what Mary Beth said, the central dogma of molecular biology is we have genes, which are your DNA, they form RNA, and they form protein. So there's a process, and things can go wrong in every single step. So that's important to keep in mind. Mutations are alterations of that genetic code, the DNA, the print that we have in our body. The other important point to keep in mind is do all Alport patients have mutations? And I think Mary Beth answered that very correctly, that do they have an identifiable mutation? You know, this is still an evolving field. And we see this not just in Fabry's or Alport. Alport is the second most common monogenic cause of kidney failure in the US. The first one is ADPKD, autosomal dominant, and we have a lot more information on that. And even in those, sometimes we cannot find an identifiable mutation, even though they're behaving as ADPKD. And at that end, I think it's really up to the provider to make sure that they have done a full investigation on the cause and they're not missing anything else. So the genotype, phenotype is actually quite important.
Thank you, Anjay. Thanks, Mary Beth. I think I do want to say if people have very specific questions about their family, we're going to take those offline and see if we can get those answered by our experts later, so we can keep this general. But Mary Beth, I do want to take, I don't know if you can see the Q&A. I want to take Jackie B's question from 12:08 p.m. and just use that as a guide to answering the question, how do we read the genetic report? And what does all that mean? In this one, can you see it? I'll read it to you.
I'm muted. Yes, I can see it.
All right, perfect. Why don't you just walk us through that and what all that means?
Absolutely, yeah. So one of the things that you see on the genetic testing report, no matter what lab does it, is you'll see the gene, and then you'll see a string of numbers and code. What does that mean? So essentially the C, like for example here, the C2083G to A, that is like a GPS code or a GPS signal for where in the gene that mutation lies. So genes are made up of DNA, which have individual letters of A's, C's, T's, and G's. And so that number, your C.2083, for example, is telling you where along that gene that mutation is, and it tells you what that letter change was or what that DNA change was in the gene. The P dot, so the P dot refers to the protein change. So that is referring to after the gene is processed into a protein. That P dot number is telling you the amino acid or the building block of the protein that changes in that protein. So that will tell you whether or not the protein is, is it made at all? Is it partially made, or is it made and just maybe can't fold properly or something like that?
Yeah, that's a great step. I think one of the things with the next question, we'll just get into the more basic. What's the first, the collagen 4A3 mean? But while you're still on it, could you explain the significance of the G to A? We see that a lot, and that often is associated with pathogenic mutations. And if you could just let us know why that's important.
So the G is referring to the DNA letter that was there on the original gene. So when we do genetic testing, we know what the call for genes look like for people without Alport syndrome. So the lab is comparing your DNA or your genetic code to what we know is typical. So we know what should be there. And when there's a pathogenic mutation that's seen, we see a different DNA letter. So Dr. Simon, you might expand on that specifically for what that means in terms of pathogenicity. But that's essentially what it means. There's a change from the G that should be there to the A.
Yeah, and so thanks, Mary Beth. And we'll see that often that G is a glycine, and that often is switched. The Alport genes have a G, it's a triplet. There's always a G and something else, and it tends to repeat. And for some reason, when that G is mutated into another protein amino acid, that causes a pathogenic variant. So that is a clue, even if they've never seen that mutation before in a previous database, that would suggest that that's going to be a mutation that might be pathogenic. Anjay, would you mind going over the very first part, the three genes, what that means and how that can impact how it's inherited and how people may be affected?
Yeah, absolutely, Jim. And thank you, Mary Beth, for that explanation, because that's something, one thing that I do want to mention before we get into the collagen is that I'm glad these questions are coming up. You should look at your report. You should read that and ask questions. And the other thing that I want to mention is that, the nephrology world, and Dr. Simon and I both are nephrologists, but we have special interest in genetics. But a lot of the people are still catching up. So you might have more information about these genetic diagnoses than even your nephrologist. So I think so how you read that will be very valuable, and you can actually, speak to your providers. So I'm glad that this came up. Now let's talk about call for A3, which I think is a mouthful, but let's break it down. So call stands for collagen. Collagen is the most common protein in your body, and it serves multiple functions. There are about 30 types of collagens available in the body, and they're coded by more than 40 genes. Now, Dr. Simon had mentioned these peptides, these amino acids, glycines, and recurring. So every collagen is made up of three peptides, and they are intertwined like a rope, as you can. So that's what call stands for. And collagen 4 is type 4. Like I said, there are different types. So this is a type 4. Why is that important for Alport syndrome? So the type 4 collagen is present in your kidneys, in your basement membrane. It's also present in your ears. It's present in your eyes. And that's where some of these symptoms are coming from. So that's a type 4. And then the last letter after that is A3. So A stands for the alpha chain. That's the chain. And there's 1, 2, 3, 4, 5, 6. In the adults, the GBM, the glomerular basement membrane, is made of three of these alpha chains: alpha 3, alpha 4, and alpha 5. And that's where A3, A4, A5 comes in. And there could be mutations, as we discussed, on different A5. The one that we think so far is the most common, but things are changing, is on your X chromosome. The gene that codes for that is on your X chromosome. It's the sex chromosome. The A3 and A4 are what we call the autosomes. And to be more specific, it's on chromosome 2. So as you know, there are 23 pairs, but this is on chromosome 2. So this going back to the central dogma that I spoke about of molecular biology. It's the genes, the DNA, then the RNA, the transcription, and then the translation into protein. So collagen is the protein after the transcription and translation of the gene. So Dr. Simon, if you want to add some more to that, but lack of time.
I think and this gets into, thank you, Anjay. It's excellent foundation there. And I think the questions come up. The next obvious question is, what does that mean for me, right? And so, and the big split is between the alpha 5 and the 3 and the 4. That's where we have our X-linked and our autosomal inheritance. And autosomal inheritance means anybody in your family, any of your children can inherit that mutation 50%. Whereas X-linked, it all depends on whether you're having boys or girls and whether you're a male or a female genetically, XY or XX. So since males are XY, and they give their Y chromosome to their sons, they cannot pass on X-linked or collagen alpha 5 mutations to their sons. But by definition, they will pass on that mutation to their daughters because they are only giving their only X, which is the Alport X. Women have 2 X's. Assuming only one of them is mutated, they have a 50-50 chance of passing that X on to either their sons or their daughters. And that is where we get into how understanding your mutations will be very important for understanding how it's going to affect your children. If you're young enough and haven't had kids yet, whether, helping decide, what the risk is for your kids if you decide to have them. I think genetic counseling and family planning is very important if you're lucky enough to get diagnosed before then. And then I think, going back to that, Mary Beth, if women have two X chromosomes and one of them's a good X chromosome, why can they get Alport syndrome?
Yeah, so women or genetic females tend to have two X chromosomes, right? So one from their mother, one from their father. When you're a female and you have Alport syndrome, one of your X chromosomes looks like this. So the Alport gene on the X chromosome has a mutation in it. The other one's okay. Something that happens in females is that for every cell in our body chooses to activate only one of the two X chromosomes. So in some population of your body, if you have two X chromosomes, your body will produce collagen from the healthy one, and in other cells, your body will produce collagen from the collagen gene with the mutation in it. And so eventually over time, you'll have a buildup of cells and a buildup of collagen proteins that have a problem in that collagen strand because some of your cells are actually making the protein from the mutated gene. And you know, everybody's different. There's nobody I don't think we have a way of predicting, you know, the percentage of our cells that make protein from the healthy collagen gene versus the one with Alport syndrome. So everyone's going to be a little bit different in the percentage of collagen that's made from the gene with the mutation versus the gene without. I hope that's getting at, Dr. Simon.
No, that's perfect. I think it's really important to note that there are no two women with X-linked Alport syndrome that are exactly the same, because the percentage of their cells that keep that Alport X chromosome active is unique to them. And so that's why some women will go throughout their life and maybe just have blood in their urine, nothing else. Others may have hearing loss, and others may go into fulminant kidney failure. And it's really important to understand that. And that goes back to the point I really strongly make in my video, that there are no female carriers. But the fact that you have blood in your urine means that you have a manifestation of the disease. And by definition, a carrier is a person that is silently carrying the gene without any negative impact. And you know, when 20% of women by the age of 60 can end up in kidney failure, that's hardly a benign state. So it's really important for women with a family history of Alport syndrome, or anybody that's getting worked up for blood in their urine and no one can figure out why, to make sure they're following through on that and get a diagnosis to make sure it's not Alport syndrome. But that's exactly correct. And the next question that I have, I kind of loaded this one. Anjay, do you have a comment on that?
Yeah, I just wanted to reemphasize the point that both you and Mary Beth made, is about these X-linked diseases. You know, and we have gone through an entire spectrum. And I will give the example of our analogy of Fabry's disease, which is a purely X-linked disease. It's only the X chromosome that is. And we have made a point to make sure that we don't call them carriers. You know, that is a wrong term. It's heterozygotes. And for all the females, and the X-linked disease classically was viewed as a disease for males. You know, females were not affected, like was pointed, but now we know that's not the case. Whatever the reason is, the random inactivation or not so random inactivation of X chromosomes, but it's important. And then it also has importance in how it manifests. There's an entire spectrum. They can have disease as bad as males can, if they're really unlucky, or they can have. So it's really important to keep that in mind as well.
So I'm going to ask you, Anjay, the corollary to that question is, why do men with X-linked end up having such bad kidney failure and go on dialysis?
Right. So I think, and I think that's one thing. So for males, they only have one X, and if the disease is X-linked, right? And keep in mind, Alport could be, you know, autosome as well. I mean, and we spoke about that, and we are finding more and more. Since they have only one X chromosome, if that X chromosome is defective, the protein that's coming out of that is also defective. So I think that's where the problem really lies, and females can have. The other thing that is also it's a kind of mutation, right? Because it could be a destructive mutation. The protein is gone completely. It's a truncating mutation, as opposed to some mutations that it's only folding. The proteins are very complex. Going back to the third part of the central dogma, these proteins are just not amino acid chains. They're folding and multiple folding, and so things can go wrong in every step all along the way. So the type of mutation also is important. So Jim, going back to the question that you had, why do especially the X-linked males do so poorly? Because they quote unquote have one. And also you have to keep in mind that a lot of other factors that control gene expression. Just having that mutation, it doesn't define everything. It's also expression of the controllers, the modifiers, the enhancers, and other things. So it's a bit more complex than just having the mutation as well. Jim. And I don't know if Mary Beth wants to elaborate more on that, but that's our understanding.
Yeah, I think that explains it really well. And this is, if we're going back to the models, so males, instead of having those two X chromosomes, we have an X chromosome and a Y chromosome. So the Y chromosome does not contain that second backup copy of the COL4A5 gene that females have. And so that's why when there's a mutation present on the X chromosome and you don't have that backup copy, that's your one copy that you have. It also explains that inheritance pattern that Dr. Simon was talking about, where, if you're a male and you have a son, by definition, typically, right, they're getting the Y chromosome. And so if you have a son, there's no Alport syndrome if you're a male and you have Alport syndrome, because you didn't pass on the X chromosome that carries it. They got the Y chromosome. Versus if you have daughters, that's the X chromosome. They're getting one X from mom, one X from dad, and they're always going to get this X chromosome.
And Mary Beth, I think just to follow this thought through on reading the genetic test results, and talking about how the mutations can affect people differently. My report shows a missense mutation. What does that mean? What are the different types of mutations mean for how badly the disease may be expressed, and why different people can manifest differently their Alport symptoms?
Yeah, great question. So when we're talking about a missense mutation, again, you're looking at the gene, the collagen gene, and you're seeing at the site or at the number of the mutation, there's a switch in the DNA code that means that a different amino acid is put there. So that can mean that, in some cases, it means that the protein that's made, so the little string of collagen, right, we're kind of using the same model, but different concept here. So the collagen string or chain that forms that trimer, which is 3 of them, that forms that rope that makes that type 4 collagen. If there's a missense mutation and the amino acid is kind of similar to the one that was there before, similar enough, sometimes the mutation is not as severe because perhaps there's like a kink in the protein. It's still being made all the way, but perhaps the shape is a little off. And so when these other types of collagen come to wrap around with it and make that chain, maybe they're just not sitting together quite the way that they would need to be to keep that function in the body and stay strong. Now, sometimes if you have a nonsense, what we call a nonsense mutation, that mutation in the DNA of the gene is, if you think about a gene as a message or like a sentence in a book, if you follow normal grammar, your period or your full stop goes at the end of the sentence. And that's how you know when you're reading that you should take a pause or that sentence has ended. So genes are similar in the way that when the body reads the code, there's a full stop in there. There's an amino acid that specifically says stop or end. And certain mutations, a nonsense mutation, will move that period or move that full stop up into the middle of the gene or the beginning of the gene or wherever along the gene that mutation takes place. And so your body is reading along the gene and saying, oh, stop. So whatever was, whatever was after that period or that full stop, the body then kind of ignores. It doesn't factor that in and continue making the protein after that point. And so what we may see is that, maybe this string, this collagen protein, maybe the body just doesn't make it at all, because whatever the gene is telling it to do is not stable in the body. So the body will destroy it and recycle it before it even gets made. Other times you may get some protein product. So again, this is our collagen chain. There are 3 of them. They wrap around like a rope. Perhaps the stop comes here. So you get the front part of the protein. So you get a short rope, but the rest of the rope is now missing because the gene told it to stop being made prematurely. So now your body is trying to make these collagen chains with a short rope. And then that is why the collagen cannot kind of keep up with the needs of the body, and then you'll get those symptoms.
Yeah, that's a great explanation, Mary Beth. And I think one of the things that I try to drive home when I'm teaching people about this is you need to have the 3, the 4, and the 5, all 3 of them to make your type 4 collagen. If you're not making a 5 or a 4 or a 3, the other 2 can't zip together and get put out into the basement membrane. So the truncating mutations or the nonsense mutations, where you have a shortened protein, those are really going to have the most severe symptoms because you're not exporting any of that collagen out to the basement membrane. And so that's going to have the most severe effects on the kidneys and the ears and the other parts of the body. The missense mutations are a little bit more variable, right? Because some of the missense mutations, the proteins may just not fold properly. They can still zip together and get out to the basement membrane. They just may not be working perfectly. Whereas other times they can't zip together. They get stuck in the cell and they can't get out. So even people with missense mutations may have different manifestations. Anjay, did you have any additional thoughts on that?
Yeah, first of all, I think this was a great explanation by Mary Beth and your follow-up, Jim. I totally agree. And just to give some numbers to this, why, once again, reading the genetic report, I think that does the same thing over and over again, the type of mutation, the question that sometimes I get asked, and one of my patients is actually on the call today. And her dad had Alport, and she has now, but her dad was never tested genetically. We tested her because she was my patient. And so do you need to get tested? The answer is yes, because, first of all, there's so many variability. Which chromosome it's lying on? Is it an autosome or in the X? And that does have implications actually for your family planning as well, which hopefully we'll get into that point. But the type of mutation is also important, so once you go past that on which chromosome it is it a missense, is it a nonsense? And it's a frame shift, it's a splicing. And why is that? So I'll just quote you some numbers, which is just corollary to what Jim and Mary Beth says. If you are a classical male 4A5 mutation, right, large lesions, nonsense, that confirms a 90% probability of kidney failure by the age of 30. 90% by the age of 30, compared with a 70% risk with a spliced site mutation and a 50% risk with a missense mutation. Now, these are generalizations. Obviously, they're within these categories, but the type of mutation does have impact, the genotype and phenotype correlation. So I completely agree what Jim and Mary Beth said. Knowing that, it's not knowing everything. There are a lot of other factors that affect gene expression as well, but it's good to know what kind of mutation you have.
And I think that's an important point to drive home about the males. If you have a missense mutation, one of my first patients I diagnosed was in his 50s. And his nephrologist didn't think it could possibly be Alport syndrome because he wasn't in kidney failure, but in his 20s. So it's important to understand that even within the classic quote unquote Alport syndrome, the X-linked male, it's very variable depending on the mutation. So we're going to get into some more in-depth questions here. And I think I'm going to start with this next one. Mary Beth, how can you get Alport syndrome if nobody else in your family has had it?
Yeah, so I think a lot of the times people think that if you have an inherited disease, it had to have been inherited from somebody, right? Our genome, our genetic profile is always changing. So I think one of the important things that I try to tell people is that we all have genetic mutations, right? I know we're really focusing on the types of mutations that cause Alport syndrome here, but it's important to recognize that our genetic profile is really fluid and it's always changing. It astonishes me that as many of us are, quote unquote, healthy as we are, because there's so much that happens during, when you're producing an egg, when you're producing a sperm, there's this kind of reshuffling of all of your genetic profile. And throughout that process, it's normal for the body to experience new mutations or for that genetic profile to have genetic mutations. It just so happens that there's a lot of our DNA that can tolerate it, that perhaps there's a backup copy of a gene elsewhere in the genetic profile. Perhaps the DNA is not a gene at all. And so a mutation out there doesn't really matter. If you want to talk about winning the lotto, unfortunately, a lot of genetic conditions will arise, just as a matter of this process of our body reshuffling itself, and you may in fact be the first person to have a mutation in that particular part of the genetic profile.
Yeah, it's difficult to understand, but they're called founder mutations, right? And all mutations have to start somewhere. And so at some point, like Mary Beth said, someone, when they're born, has the wrong mutation and develops Alport syndrome. And the statistic that I've seen, anywhere between 15 and 30% of patients with Alport syndrome do not have a family history when they're first diagnosed. So that's an important question, and great, thank you. The next one, I'm going to leave up to whoever knows the statistics on this. I will say I have seen African-American patients with Alport syndrome, but does anybody know the rate, Caucasians, African-Americans, Latin patients, does it affect people equally across all ethnicities?
I think if Mary Beth wants to take that, as far as I know, we don't know any difference, Jim. But that's.
I haven't seen any particular evidence that certain populations are more frequently affected.
And also,
yeah, go ahead. Go ahead, Anjay.
No, I was just going to say that you also have to keep in mind it's such a rare disease. It's an underdiagnosed disease. In the US, we have all these. And I know there are some of the questions coming up for broad panel testing. And I do want to get our view, our three people who are on the panel, about the broad panel testing. But we are just at the cutting edge front of this genetic testing. So as we get more data, we might see differences, right? And that's where I think all these registries are important and contact. But as far as I know, and this same applies for Fabry disease too, by the way, I always give that comparison, but we don't see any ethnic or racial differences as far as we know.
And I think this goes to, a broader concept of disparities in healthcare, not only in America, but around the world.
Absolutely.
It was started in I think the strongest genetic registries are in Europe and the US, but I will say we have China has a very large Alport community that ASF works with. And as a lot of other countries develop, as genetics becomes cheaper, depending on their healthcare states, you're going to find that a lot of other countries will be diagnosing this as well. So there is not necessarily, none of us have seen any preponderance evidence to say that it does not affect African Americans the same as other ethnicities in the U.S. or around the world. I have one more diagnosis question, and then I think a lot of it's going to be going on to the genetics, and we'll get into the broad and some of the other questions, Anjay. So the last one, I think, before we get into the deeper into the genetics, is, does Alport syndrome affect kidney transplants? Anjay, I'm going to let you explain that.
Yeah, so that's a very, very good question, actually. Technically, no, because when you get transplanted, your kidneys come with their own new genes, and that does not carry the defective gene, and that applies for ADPKD, all the other monogenic causes of kidney disease as well, Fabry and stuff. But you really have to keep one thing in mind, and that's something your transplant people will talk about. That is about antibodies to your GBM that might be present, and that might cause rejection. So it happens in a very small percent of cases, but can happen. So that's your body reacting to something that they have not been exposed to in the past because they never had that. So that's the only thing, Jim, that I would say would be effective. But that the transplant program that you're going to will be discussing with you in any case.
Okay. Mary Beth, I'm going to throw a couple of questions at you. What are frequently asked questions that a patient should be prepared for when going to see a genetic counselor, and what should they expect out of the genetic counseling session?
Great question.
Yeah. So frequently asked questions during a genetic counseling session, most commonly we'll start off the session by really asking what it is that brings you here today as the patient. We want to make sure certainly that we get certain information across, and we ask a good thorough family history. So if you know that you are coming to a genetic counseling session and you have the ability to talk to your family members about their own health, then that's good information to have, not just about kidney disease or hearing loss, but maybe anything else in the family tree that we may learn has a genetic link, so that we can make other appropriate referrals. For example, early breast cancer or ovarian cancer certainly come up quite a bit. So first of all, we're going to ask what it is that you want to learn and what your motivation is for coming to a genetic counseling visit in the first place, and then try to tailor our session around that. And what was the other part of the question? I'm sorry.
Oh, just what questions should they come prepared with, and what do they expect to get out of it?
Sure. So I think a lot of it depends on where you are in the life cycle, right? And where you are in your journey of diagnosis. So if you're the first person in the family to have kidney disease, maybe we haven't even done genetic testing for you yet, and we're still trying to kind of determine, is this Alport syndrome, or is this something that could mimic it? So I think if you're, an adult and you're coming into a genetic counseling session and you've just learned that you have kidney disease, your questions are going to look very different than if you're a parent who's coming in and you've had a longstanding diagnosis and you're wanting to know what those risks are, or what those chances are that you're going to have a child with Alport syndrome. So think about, if you are being referred to genetic counseling, kind of think about where you are in the diagnostic process. Is genetic counseling and genetic testing going to be helping you find that gene, and you're the first person in the family to have that confirmed? Or is the genetic mutation known and you're planning a family and you want to have some help talking to your partner about this and kind of figuring out where to go from there? That's going to be where you're seeing a prenatal genetic counselor versus a general genetic counselor and that type of thing. But really, the visit is designed to be tailored around what your specific needs are. So don't feel like you have to have even an agenda to come in. We can help to figure that out.
We have 3 minutes left, actually 2 now. I'm going to take one question really quickly, and it's if I already have someone in my family who is genetically diagnosed and I'm showing symptoms, do I need a genetic test to confirm it? And I will say that's very specific to the person who's taking care of you. Some people are comfortable telling you that you have Alport syndrome, and others, especially in the pediatric nephrology population, will want to get you gene tested. So that is not a uniform question across the Alport community, even amongst the experts. The last minute and a half here, I'd really like to talk about autosomal dominant Alport syndrome and just why that's a kind of a different beast of its own, why some people might not have any symptoms and others can go into kidney failure. And is there any concern with women and getting pregnant with autosomal dominant Alport syndrome? Who wants to take that? We got about a minute left. No pressure.
Mary Beth, you can take a half a minute crack on that, and then I'll give you my opinion on that as well. There is no right answer for that. It's an evolving field, this autosomal dominant.
Yeah, and I think that's a great question. I'm actually going to throw that over to you for the whole time.
Yeah. So let me tell you this autosomal dominant. I mean, we have X-linked. That's probably pretty clear cut. We also have autosomal recessive. You know, you have two genes, and they behave just like, you know, an X-linked male would. But the autosomal dominant is really, in my opinion, you know, for lack of better words, is an evolving field. And if you talk to genetic counselors, you talk to geneticists, people who actually do this for a living, even they cannot answer that correctly. There is something that we call thin basement membrane nephropathy. Sometimes we call it disease. Sometimes we call it or benign hematuria. So my short answer of that, if you are being diagnosed with autosomal dominant type, or that comes in your gene report, the genotype phenotype correlation is very, very important. You need to be followed very, very closely. And I, for this one person, would make sure that you are talking to a geneticist, you know, who is also and working in collaboration with them. So the genotype is going to be very important in this case. And this could be, in a lot of cases, just a benign, you know, but we don't know that yet. You know, it's very difficult to just say clearly. But there are autosomal dominant in which it does progress.
Yeah, great. Thank you. Lisa, I know we're getting the hook here. And if you want to tell the audience how to address the questions that have been asked that we couldn't get to.
The questions that we couldn't get to, I did take pictures of to make sure that we know we can get back to you. And we will send along your questions if they weren't answered here. We'll send along your questions, panelists or appropriate experts.